Hi guys, I'm Catherine and today I'll be talking about vision. So first of all, vision is actually one of your most complicated senses because it involves many different processes that work together to enable you to see what's happening around you. Actually, the visual system involves around 30% of your cerebral cortex, which is more than any other sense. Furthermore, vision has been studied intensively using fruit flies, mice, monkeys, and cats. In many ways, seeing with your eyes is similar to taking pictures with an old-fashioned camera. The light passes through the cornea and enters the eye through the pupil. The iris regulates how much light enters by changing the size of the pupil. The lens then bends the light so that it focuses on the inner surface of your eyeball, the retina. The cornea does the focusing, and the lens can thicken or flatten to bring near or far objects into better focus. First of all, I'll be talking about the retina. The retina has three types of neurons: photoreceptors, interneurons, and ganglion cells. These are all organized into different layers. The light-sensitive photoreceptors include rods and cones, and they are located in the most peripheral layer of the retina, or the back. Light travels through the ganglion cells and interneurons before it reaches the photoreceptors. Ganglion cells and interneurons do not respond directly to light, but they process and relay information from the photoreceptors, and the axons of the ganglion cells exit the retina together and form the optic nerve. There are around 125 million photoreceptors in each human eye, and they turn light into electrical signals, which is known as transduction. Rods make up around 95% of photoreceptors in humans, and they are extremely sensitive, so they allow you to see in dim light. Cones pick up fine detail and color, and they allow you to engage in activities that require a great deal of visual acuity. There are three different types of cones that are each sensitive to a different range of colors: red, green, and blue. Because their sensitivities overlap, the different combinations of the cone's activity convey information about every color, and they allow you to see the color spectrum. The center of the retina contains many more cones than the other areas, so vision is sharper here. In the very center of the retina, there is the fovea. The fovea is a small pitted area where cones are the most densely packed. It contains only red and green cones, and they can resolve very fine details. The area around the fovea is the macula, and it's critical for reading and driving. Macular degeneration is the death or degeneration of photoreceptors in the macula, and it's a leading cause of blindness. The amount of inputs varies widely across the retina. The portion of visual space providing input to a certain ganglion cell is called its receptive field. In the macular region, the receptive field is small, so the visual acuity is the highest. Each ganglion cell receives input from just one or very few cones, allowing you to resolve very fine details. Near the margins of the retina, however, each ganglion cell receives signals from several photoreceptor cells. This convergence of inputs explains why your peripheral vision is less detailed. Now you may be asking, how is visual information processed? Visual processing is how you distinguish everything that you see. It begins with comparing the amounts of light hitting small adjacent areas on the retina. The receptive fields of ganglion cells tile the retina, providing a complete 2D representation or a map of the visual scene. The receptive field of a ganglion cell is activated when light hits a tiny region on the retina that corresponds to the center of the field. However, when light hits the donut-shaped area surrounding the center, the ganglion cell is inhibited. If light strikes the entire receptive field, then the ganglion cell responds weakly. This allows you to maximize the perception of contrast, which is the key to object detection. The optic nerves transmit the neural activity in the axons of ganglion cells. The information exits the back of each eye and travels towards the back of the brain. Because there are no photoreceptors, the exit point of the optic nerve results in a small blind spot, which our brains fill in using information from the other eye. On their way to the brain, signals travel along nerve fibers from both eyes, which first converge at a crossover junction called the optic chiasm. Information from the left side of the retinas of both eyes continue to the left side of the brain, and information from the right side of both retinas go to the right side of the brain. Visual information is then relayed through the lateral geniculate nucleus, a region in the thalamus, and then to the primary visual cortex at the rear of the brain. The primary visual cortex is a thin sheet of neural tissue located in the occipital lobe in the back of your brain. This region also consists of many layers with densely packed cells, like the retina. The middle layer receives messages from the thalamus, and it has receptive fields similar to the retina, and it preserves the retina's visual map. Cells above and below the middle layer have more complex receptive fields, and they register stimuli shaped like bars or edges, with particular orientations. From these layers of the primary visual cortex, new processing streams pass the information along to other parts of the visual cortex. As visual information from the primary visual cortex is combined in other areas, receptive fields become more complex and selective. Some neurons at higher levels of processing respond to only specific objects and faces. Then, visual signals are fed into several parallel but interacting processing streams. The dorsal stream heads up to the parietal lobe, and the ventral stream heads down to the temporal lobe. Crosstalk between streams actually create a conscious experience. The image is recognized by the brain. It extracts information at several stages, compares it with past experiences, and passes it on to higher levels for processing. Binocular vision is seeing with two eyes. This allows you to perceive depth, which is important, because each eye sees an object from a slightly different angle. But this only works if the eyes are equally active and the visual fields overlap. Also, two eyes allow a much larger visual field to be mapped in the primary visual cortex. Because some of the nerve fibers exiting each eye cross over at the optic chiasm, signals from the left visual field end up on the right side, and vice versa. There are many visual disorders, and strabismus is a common one. Strabismus, or being cross-eyed, is where children have good vision in each eye, but because they cannot fuse the images coming from both eyes, they start to favor one eye and often lose vision in the other. Vision can only be restored only if the child is treated at a young age below 8 years old, or else the blindness will become permanent. Blindness is caused by a loss of function or death of photoreceptors. Unfortunately, it is very difficult to treat. Genetic studies have identified a variety of genetic defects that cause people to go blind, so gene therapies or stem cell therapies can recover photoreceptors. Another solution is sending electrical signals directly to the brain via ganglion cells rather than attempting to restore the lost photoreceptors.